at Spelman College in Dallas, Texas. Under the past 2 semesters, I have worked with Dr. Davenport at Morehouse College studying Leishmaniasis cells and their treatment methods. Leishmaniasis is a parasitic disease that typically infects tropical regions, including Africa, Asia, and parts of Southern Europe. The disease can cause skin lesions while also affects our visceral organs, leading to other complications. So it's very important to find a effective treatment method compared to the ones seen today that are either very costly or toxic to our bodies. And so in our lab, we are specifically studying the visceral form of leishmaniasis, which is the most deadly, and we will study the apoptotic methods of the cells. Apoptosis is induced cell death, where the cells commit suicide. And so we are trying to see if we can manipulate that into a treatment method. But before we can do that, we first have to confirm we are seeing apoptosis. So within this past semester, I studied the effects of hydrogen peroxide on the Leishmaniasis parasites. And hydrogen peroxide is known to be an apoptosis inducer. However, we have to confirm. And so within my studies, I first analyzed the effects of that through fluorescence microscopy. And so with the calcine stain we see here, it should highlight green. And with only calcine, we can see that that specifically highlights here the early apoptotic stage. But with propidium iodide, that highlights specifically necrosis, otherwise known as regular cell death not induced or self-suicide. And so with that, as it highlights red, that highlights necrosis. And when both overlay together, we can see it specifically being the late apoptotic stage. And so I will continue my research with image cytometry and flow cytometry to not only see the cells, but to count them to see how many are actually viable, how many are just dead, or how many faced apoptosis. And so with that, I will continue my research after confirming apoptosis in the lab to see the effects of membrane permeability from apoptosis, to see how we can manipulate that into a treatment. Thank you for listening and